Hello, I'm Nathan Chapelle, a first-year PhD student from the Physics Institute of Rennes, where I'm doing my work under the supervision of François Potserf and Isabelle Cantat. And I'm working on how the bacteria in a soil can change the drying dynamics of this soil when they release their surfactants. So, evaporation of a soil is a key hydrological process, and it is a microscopic phenomenon. But this microscopic phenomenon is governed by some capillary flows along water films. And on this plot I took from the literature, what we can see is that as long the water films remain stable between the water table and the top of the soil, we are on a stage where the evaporation process is quite effective. However, when these films break, we are now in a stage where the evaporation process is much less effective, and the idea of my PhD is to shed light on how the bacteria in a soil, when they release their surfactants, can destabilize these films and lead to a change of stage at a microscopic scale. So to do so, I'm working with a soil model bacterium, Bacillus subtilis, which produces a powerful biosurfactant, surfactin. And so far, I'm working at the pore scale. So I built a microfluidic device which mimics one pore of the soil. And in this pore, I'm trying to understand how the surfactants, when they are released, accumulate at one air-water interface and can change the properties of this air-water interface. So first of all, I made some experiments in order to characterize the release of surfactants by Bacillus subtilis. To do so, I used two different strains. In dark blue, it is a wild-type strain, so this strain releases surfactants, while in light blue, it is a GMO strain which cannot produce surfactants. What we see on this plot is that during the growth of these two strains, the wild-type strain strongly decreases the surface tension of the broth, while the other one doesn't change the surface tension at all. It means that Bacillus subtilis can decreases the surface tension in the middle where it grows, and the interesting thing is that this effect can occur with a relatively small amount of surfactin. For instance, the concentration of surfactin here is like 100 micromolar per liter. So to understand how these surfactants can change the drying dynamics in one pore, I'm using this experimental setup. So I've got one chip with, at the top of the chip, one air-water interface with a meniscus here, which is pinned on a sharp edge. At the other end of the chip, I have a reservoir which is connected to the chip, and this reservoir is pressure controlled to have a fixed water depth during the evaporation process. Because of the evaporation at the top of the chip, there is a water flow in the chip, and this water flow implies an accumulation of surfactant toward the air-water interface. I'm monitoring the air-water interface thanks to a microscope, and what I see is that during the accumulation of surfactants, the meniscus deforms, and to predict this deformation, I'm using a 1D transport model based on an advection diffusion equation, where the advection term is directly linked to the evaporation rate. I solved this equation with my boundary condition, and here we can see that at the air-water interface, the concentration increases over time. And so the next step is to link this change of concentration just under the air-water interface to the surface tension to be able to have a prediction of the deformation of the meniscus and to compare this prediction to the one I see in my experiment. Thank you very much. See you.